Broadcast is now starting. All attendees are in listen-only mode.
Hello and welcome to today's webinar. On behalf of European Pharmaceutical Review and B&W Tech, a Metro Group company, I'd like to thank you all for attending. I'm your moderator. My name's Nikki Withers and I'm the editor of European Pharmaceutical Review. Today's speaker will be Dr. Catherine Bakayev, who is the Director of Products at B&W Tech. Following her presentation, we will move on to a live question and answer session where you can pose questions to today's speaker. Please remember you can submit questions at any point during the webinar using the questions panel, which is situated in the menu on the right. So without hesitation, may I pass you over to today's keynote speaker, Dr. Catherine Bakayev.
Thank you, Nikki, and thank you everyone for joining today. Today I'll be talking about quantitative transmission Raman for content uniformity testing. Specifically, I'll be talking about developments at B&W Tech for the QT-RAM instrumentation for the quantitative transmission Raman. But first, let's begin with what is Raman spectroscopy. Many of you may have used FTIR and studied in school, and hopefully some of you have also done the same with Raman spectroscopy. It is a form of molecular spectroscopy complementary to FTIR, wherein we use an incident light, a laser, to excite the molecules, and there is some that undergo an energy shift, which is then collected in terms of the scattering light of and this is what we call the Raman scattering, the energy that undergoes a change in energy or on exciting those molecules. And from that, we get specific peaks related to the different chemical functional groups. And this gives us then what we call the molecular fingerprint. The Raman spectrum is considered a molecular fingerprint, and we can use that to identify a material. From this, we can also get information about the polymorphic forms of materials. We can get quantitative information. So a larger concentration of materials, the stronger the peaks that we have, the position of the peaks related to what the chemical makeup of the system is. So Raman spectroscopy has a great advantage of giving us so much information with direct measurements on samples. We don't need to do sample preparation typically. It is non-destructive, and we can measure through clear containers and transparent containers to measure that Raman spectrum. It's quite a rapid method by which we can get information about multiple chemical components in a system all at once. There are many different areas across the pharmaceutical manufacturing where Raman spectroscopy can be used. From the beginning of the incoming raw material verification using handheld Raman spectroscopy typically through to the final product release, and also even in counterfeit detection. Many a company has Raman spectroscopy in their teams where they are getting materials that may have been seized and determining whether it's the authentic product that they have made or if somebody is counterfeiting their products. As we look across the manufacturing process, then we see, yes, the raw material identification can be done, but with Raman spectroscopy, we can also apply this to the chemical syntheses, the crystallization processes. These are fiber optically based systems. They can be interfaced into processes to follow the process in real time and get information on the chemical transitions and changes. This applies as well to the dry processes, to the milling, granulation, and drying. And then we can do this as well after the tableting, so we can get the API content, the content uniformity of the active pharmaceutical ingredient, so important for the quality of the medical products. So what we also can do is then do some testing on the final products, verifying that they are what they're meant to be, verifying coating thickness, and as I said, product verification. So as we talk about this, let's now go to a poll and see how it is that maybe you use Raman spectroscopy across your company. It is complementary to FTIR, so many a laboratory uses both to get the full information, the chemical information about materials. But there are other technologies as well that may be being used. So we'll give it a few more minutes to let everyone give us their responses, and then we'll continue our discussion on quantitative Raman. Okay, well, we see that there are people that use the Raman in a lot of different ways across their industry, so why don't we wrap up this poll and take a quick look at those results. And what we can see is that many of you are using actually Raman within R&D and for incoming raw material identification, but equally as many are currently not using neither Raman nor near-infrared spectroscopy. So hopefully after learning some things today, you'll see how this can benefit you in your process. So today, what our focus is, again, is on the content uniformity using transmission Raman spectroscopy. So the other application areas, we have many application notes on, and we're happy to talk to you about, but for today, we're focusing on our content uniformity testing. But one of the first things to consider is, why do we use Raman? Well, as I noted, across the manufacturing process, there's a lot of good information we can get, and it is an important tool for ensuring the quality of our products and the quality of our medicines. Because it's a non-destructive and rapid tool, we can use it, as noted, from formulation to the final product QA, and diminish time for our testing and even for our product release. As many of you are aware, pharmaceutical formulations are comprised of different components, and with one Raman spectrum, we can get the information to measure the multiple ingredients from that one measurement, which may be just a matter of seconds to measure. Of course, content uniformity is very important because it is required testing for solid dosage forms. And the traditional methodologies, the HPLC, require the samples to be consumed, and they are time-consuming processes and generate waste as well. Whereas with Raman, we can measure the tablets as they are without doing any further sample preparation and not requiring any other consumables in that regard. So we use Raman for tablet analysis because it is rapid and non-destructive, and it gives us sharp, distinct peaks. It's a highly selective methodology. What I have shown here are the near-IR spectra of tablets. Near-infrared has been used for tablet analysis for over a decade, whereas Raman is used in tablet analysis, and transmission Raman is coming to the forefront now. So with Raman analysis, we can measure through the tablet coatings, and as opposed to near-infrared, we don't have a strong contribution from water, so the signal isn't masked by water. So we don't have interferences from there being varying moisture contents of the samples that are measured. Now, transmission Raman is an important advancement because in traditional Raman spectroscopy, as shown here on the left, what we're doing is a conventional backscattering measurement. So the Raman laser excitation, goes onto the sample, and we're measuring the Raman light that is scattered back. So our measurement is close to the surface. With transmission Raman spectroscopy, we have the light transmitting through the body of the sample. So we get a volumetric sampling as that laser light diffuses through the sample and the scattering is collected on the other side. So this provides a representative measurement of the sample's content beyond that near surface measure of the conventional Raman. But there are some challenges with Raman spectroscopy. One is that the laser beam and the Raman beam are both focused, so we tend to have a very small sampling volume. And this undersampling can lead to irreproducible results. So in the typical case, we are looking at a very small area. And if we go from one spot to another, we may get spectra that look different. And what I have shown here are actually the spectra taken at various locations of an over-the-counter medication that has 4 different ingredients in it. And we see that the spectra don't perfectly overlay each other. We can overcome this, and we have done this with a design in our instrumentation by defocusing our light to get a larger sampling area. And when we do this, then measuring this same over-the-counter medication, we see that with over 30 spectra measured, they are perfectly overlaid with the other. So what we have is a patented reflective cavity, which increases the sample size and the sampling volume while keeping our signal strength. So really the benefits of Raman spectroscopy, and in particular in the transmission Raman, is that we are being able to penetrate and transmit through the sampling volume. And we can quantitate numerous ingredients all from one scan. Likewise, we could also get the complementary information on the structural form, the polymorphic form, or the crystallinity. And this can all be done at line, further increasing our analysis time turnaround. It should also be noted that Raman technology is recognized by regulatory agencies, being included in the different pharmacopoeias. And just now in August 2020, the US Pharmacopoeia has released 2 updated chapters, or I should say new chapters replacing the original, chapters 858 and 1858 for Raman spectroscopy, really pointing out the acceptance as well as the importance of Raman in the pharmaceutical industry. So this is showing the QT-RAM system and our patented design, wherein we have separate excitation and collection pathways of our light. The design is such that we have our cavity here, wherein we are collecting numerous reflections of the light so that we are increasing our signal not just in the sample's size and area, but also the strength by collecting the multiple reflections of that Raman light to get a strong signal. With our transmission Raman with defocused light, we are overcoming some of the inherent obstacles of transmission Raman. The first of which is typically the signal is quite low as the light has to pass through the entire sample volume. But with our reflective cavity, as well as using a high throughput spectrometer, we get very good signal. Likewise, in most solid dosage forms, we have numerous excipients which may cause fluorescence and reduce the overall signal-to-noise ratio of your Raman spectrum. Our signal enhancement techniques allow us to overcome this challenge. Another obstacle is that it is considered non-trivial to do transfer of quantitative models from one instrument to another. The key to this is to have highly reproducible and consistent instrumentation, and we manufacture our instruments with this high consistency, as well as the necessity to develop robust calibration models that do not model anything, noise and things like that, but are really relying on the true signal of the samples in the modeling. So if we look more closely into the QT sampler, what we have is tablet holders, and these are made to be round or oblong with a maximum tablet thickness of 10 millimeters. And they can be customized to the dimensions of the particular tablets of users. This tablet holder is then placed into the QT sampler, where we have excitation and collection apertures of different sizes. The sample holder is placed on top. The apertures also are of different sizes, so that if you have a larger sample, you are measuring over a larger size to get the best representative sampling. With the sample in place, the stage is then closed to begin the measurement of the transmission Raman. So now I'd like to walk through a case study of using transmission Raman spectroscopy for low-dose API content. And in this work, we use the QT-RAM with the BW Analyst software, and the software is on the embedded touchscreen, so it's touch-friendly software, and follows a logical workflow for the method development. So when we go into the method development module of the software, we have step-by-step workflow where we begin by acquiring our calibration spectra, and these are of samples that are then sent for HPLC analysis, because as with near-infrared spectroscopy, we need to develop our calibration models from samples of known concentrations. So with our real samples, we want to get those reference values, typically by HPLC, but you'll see in this case study, we used gravimetric samples. They were specially prepared using a design of experiments. So we acquire the spectra, export those data to develop a model in our chemometric software. The partial least square regression model is then imported back into the software, a method built around it where we enter the information such as the tablet weight. We validate that model, and then we can release the method. The method validation requires that we also have acquired validation spectra, which is one of the steps in our flow here. Those can be collected at a different time or at the same time that our model spectra are collected. So we began this work with a design of experiments, which is always a good way to develop a calibration set. And actually this design of experiments in the tablet manufacturer was done at Rutgers University, and we thank them for their support in this. So defining the calibration samples, we had the constraints that we wanted the lubricant of magnesium stearate to be at a constant 1%. We wanted carmellose, one of the excipients, to be at less than 10%. And we wanted to have a very low correlation amongst all the ingredients. So blends were made of 5 different components: APAP or acetaminophen, mannitol, silicified microcrystalline cellulose, cross-carmellose, and then the mag stearate at the constant 1%. So if we look at this in a design triangle, we have a small portion of this where this is. And we had samples then, 9 different blends ranging from the active concentration from 0 to 3%. And this is to show that we do not have correlation of the active ingredient with any of the other ingredients. And this is important because in model building, we want to make sure that we are actually developing a calibration model where our correlation is to the signal of our active ingredient and not relying on signal of other things which we are not looking to quantify with that particular calibration model. So our data acquisition was done after preparing samples from the blends. They were pressed into 10 millimeter diameter tablets of 3 millimeter thickness, each of them a nominal 300 milligram weight. And our target dosage was to have 1.5 milligrams of the APAP, acetaminophen, which would be 0.5 weight %. So the data were collected on the QT-RAM using 4 millimeter diameter aperture, and we loaded up 8 different tablets at each one of the concentrations and collected numerous scans per each sample. When you measure your sample, you can load the samples and actually measure on both sides by removing the sample holder and rotating it around to the other side, which is something that we recommend. So this is showing all the raw data collected over all the calibration samples. Now, you may recall that our calibration samples have samples from 0 to 3% of our acetaminophen. And this is showing us how good the Raman spectroscopy is for the materials that we're wanting to measure. And this is a very good first step before developing a calibration model, is to look at the Raman spectra of each one of the ingredients with that of the material, the blended material. And here we see that we have very strong signal from our mannitol, one of our excipients, from our APAP, the active ingredient for which we want to quantify, and the microcrystalline cellulose. And we have it marked here where we have distinct peaks that are separated from the other materials in the formulation. And we don't show the cross-carbamylose, which has mostly broad bands and fluorescence, nor the magnesium stearate, which is too weak to discern visually in the Raman spectra. Now we note that, again, we have very strong signal from the acetaminophen, and we will focus our calibration model on this range up to about 1700 wave numbers, even though we are collecting our spectra all the way out to 2500 wave numbers. So we start by developing a calibration model over the full range of the sample concentrations that we have, and we see that we get quite a good linear calibration model. But our real interest here is to be able to quantify at the target concentration of 0.5% of our active pharmaceutical. So in addition to our full wide range model, we also developed a low dose model from 0 to 1.6% of the active ingredient. And we did this because by separating the ranges, we can increase the accuracy at this low dosage. You see that our error of calibration is 0.02 in the more targeted model, as opposed to 0.045 in the wider range model. And by looking at the data a little bit more closely as we do our modeling, what I have shown here on the left are the first derivative spectra of the active ingredient that we are quantifying, as well as the first derivative of one of the main excipients, the mannitol. And these then are the loading vectors from our partial least square regression model. And we see very closely how the first loading, which explains about 71% of the variance in our calibration data set, very much follows the signal and carries the signal of the active ingredient. And we see this also in the scores plot shown to our right, where the markings around each one of the circles are the first number is the APAP concentration, the second the mannitol. And as we look at that plot from the left to the right, we see that we go from the low concentrations of the APAP to the higher concentrations. Again, this is verifying that indeed, and another visualization, that this first factor explaining about 71% of the variance of our data is related to the signal change with the APAP. Likewise, our second factor, second loading variable, has signal that very much resembles that of the mannitol. And if we look at our scores plot, the map of our samples in our multivariate space, we see that we go from the low to the high concentration of the mannitol along the factor 2 axis. So this is a very good reassurance that our correlations and our modeling is related to what it is that we are wanting to quantify, our APAP concentration. The next thing that we want to do is to validate our model, and we validate this with the validation samples that we've measured, and we see here that we get very good predictions of our samples, and we validated the low concentration as well as samples near 0.5%, and our prediction is quite good here as well. And from this, we're able to compute our limit of detection as well as our limit of quantification. And we see that these are quite good with our LOQ at 0.23% of our APAP. So we see that this transmission Raman method works very well for the measurement of the APAP across the concentration range of 0 to 1.6%, which is what our calibration model is. Now, we had developed 2 calibrations, but we are looking at the lower concentration. But we wanted to see how well this model at the lower concentrations could also predict samples outside of that range. This is not something that would be the way to validate our model if we were only using it in the small range, but we wanted to see, again, how robust our model is. And yes, we are able to predict samples outside the range as well with this model. Though again, this wouldn't be recommended to use models outside their calibration range. If we were going to be running samples at the higher range, we would want to use our wider range calibration model. But as our target is actually at the 0.5%, we are using the model spanning from 0 to 1.5% of our APAP. Another thing that we wanted to test was the transferability of our model. So this calibration model was built on one QT-RAM instrument, and then we took those data and that model, loaded it onto a second instrument, and collected additional validation samples on that second instrument. And we see that we have a very good linear fit of that model that we've transferred. We have a very small transfer bias and a small root mean square error. So we see that the transmission models are transferable even when we're at this low concentration and when our data have fluorescence in them as well. So this is a validation of the fact that transmission Raman can work very well even at these low dose concentrations of active ingredient, and that we can transfer the model from one instrument to another. So I hope that I've been able to show you that Raman spectroscopy is a valuable and non-destructive tool that can bring important information throughout pharmaceutical manufacturing processes, and specifically to content uniformity testing. Using transmission Raman for solid dosage forms gives representative sampling because you're measuring across the volume of your sample and not just near the surface. And from that, you can measure the active pharmaceutical ingredient content, as well as the concentration of other ingredients in the solid samples, even at low dose levels. So though I've only shown you the calibration for the APAP, we've also developed calibrations for the mannitol concentration in these samples as well. And so with one Raman spectrum, we can predict the concentration of the mannitol as well as the API in the tablets in less than a minute. For the calibration model that we've developed here, we have a limited quantification, which is well below 1% for the APAP, in a matrix where we have relatively high fluorescence interference. We've also shown that direct calibration transfer is possible when we have consistently built analyzers and robust models as we've developed here. And I'd like to acknowledge the development work, modeling, and analysis by my colleagues at B&W Tech, Jun Zhao and Chris Kautz. Support in data collection and sample preparation from Professor Rodolfo Romanach's group at the University of Puerto Rico, Mayagüez, as well as Dr. Andres Roman at the Rutgers University, the State University of New Jersey. So with that, I'd be happy to answer your questions at this time.
Thank you so much, Catherine, for that excellent presentation today. And as you say, we now have the pleasure to introduce our questions and answer session. where Catherine will be able to answer all of your questions. Just a reminder that you can still submit any questions using the questions panel in the menu on the right-hand side. So let's take a look at the first question. Catherine, what is the typical analysis time for the Raman?
The typical analysis time is under 1 minute, but it is sample dependent, and yes, you can also do some averaging to increase your signal to noise behavior, but the typical analysis time is under 1 minute.
Lovely, thank you. So our next question is can liquids be measured?
Liquids can be measured by Raman spectroscopy. The design of this particular instrument is not to measure liquids. It is designed for tablets, but let me actually go to a different slide and show you another configuration wherein liquids could be measured. So if you'll recall here, I've shown a couple of different instruments here, and let me get back to a good view here. So in all of these cases, these are fiber optically coupled instruments. And here I have it coupled to a microscope. It can also be coupled to use with cuvettes or disposable glass vials, as can our handheld instruments, the NanoRam instruments. So the transmission Raman of the QT-RAM of this instrument is designed for solids. The sample holders are for solid dosage forms, but Raman spectroscopy works for liquids as well as solids.
Lovely, thank you, Catherine, for clarifying that one. Our next question is how many samples are needed to develop a calibration?
That too depends on the number of, well, let me say you would need at least 15 samples spanning the concentration of interest for you. So it's best to have samples. If in the case that we just showed, we had 9 different concentration samples, but we had numerous tablets of each concentration. So you would want to have not just many samples at one concentration, but several samples at a number of concentrations across the range. And I would say a minimum of about 15 tablets.
Perfect. Our next question that's come in is, can different tablet sizes or shapes be measured?
Yes, they can. The tablet holders, as I've shown you, the oblong one can fit different sizes, but we also have different size holders that can be used. So yes, they can be. It's advisable that within one calibration model, the same dimension of tablets be used, especially the same thickness.
Great. Our next question, can concentration of multiple ingredients be predicted from a single Raman scan?
Yes, that can be the case. Here we show the concentration of one of the ingredients, the APAP, but we have developed for this exact sample set an additional calibration for the mannitol. When we have multiple ingredients that have a Raman signal, as we showed here in this formulation, then we can develop calibration models for the different ingredients within them. Yes.
Lovely, thank you. And our next question, is transmission Raman spectroscopy accepted for pharmaceutical final product testing?
Yes, it is. Yes, it is accepted, and there have been companies that have been developing and using this as final release, a transmission Raman spectroscopy in that manner.
Great, thank you for clarifying. So our next question, you mentioned signal enhancement techniques. Are you able to provide more information?
Yes, let me go back to that, our signal enhancement. So if we look at this, we can, there are some publications that we have in that, as well as our patent on this, but the idea is that indeed this is a highly reflective material. It's actually gold coated within the holder. So we use high throughput system and a larger throughput as well, larger fibers to get more light going in. But we also get the signal enhancement, by having the light that's reflecting off the sample recollected as it bounces off, and then on the collection side as well. So we're collecting as much, pushing as much light through and collecting as much as we can too by measuring these multiple reflections.
Great, thanks for clarifying. The next question, well, it is actually a question. So operation software and chemometric model development software are different. So I think that's asking whether they are different there.
Yes, they are. In this system, yes, we do have the BW Analyst, which is the operating software on the touch screen, and we have a chemometric software, BW IQ. Yes.
Lovely, thank you. So a question about percent variation. So what is the acceptable percent variation for results between Raman spectroscopy and other primary methods like HPLC, CUT testing?
Well, you know, a method becomes usable after you've validated it. So it's important, first of all, that the values, the reference values that one gets from HPLC are highly accurate. Because that will, if they are not, then that will introduce error into a Raman method. But, you know, one expects that with good and accurate reference values that your Raman spectroscopy method can match that type of accuracy in the methods.
Lovely, thank you, Catherine. This question has come in, hopefully I'll be able to word it right for you. So the main question is, why is the validation set needed to be run on a second instrument during method transfer? And as a follow-up, saying that we cannot transfer the method from, can we not transfer the method from one instrument to another?
So the question is then that I stated that we transferred the method and then we validated it with additional validation samples on additional instrument. Well, that would be good practice actually to validate a method once you've transferred it. And in order to validate it on the other instrument, then the samples need to be measured on that instrument.
Right, thank you for clarifying that for me, Catherine. Is the software validated for use in my QC lab?
The BW Analyst software is 21 CFR Part 11 compliant software.
Lovely, thank you. Going down to our next question. Have you tried the instrument for API quantification in capsules?
I have, and unfortunately I don't have the data in front of me, but there are some capsules that it's a little bit more difficult for the light to penetrate. So it does not work for every capsule, I have to say, depending on the capsule thickness. The important thing here is that our light can get through the capsule material and then through it on the other side as well. And depending on that gelatin material and the color of it, that can be challenging. So I can't say that this will work for all capsule formulations.
Okay, thank you. Thank you everyone for sending these questions in. We've got a few more to go through. So how much are results affected by variability of tablet dimensions?
So because we're doing a transmission measurement, we should keep this dimension similar, because this will be related to how much light and how much material we're sampling at the same time. But if our sample, if the diameter is different, that should not be problematic, because our spot size will be the same. So it is really the thickness dimension that would be a challenge and would introduce variability in the measurements that you would want to minimize.
Great, thank you for confirming that. Our next question, does Raman spectroscopy require sample preparation, or can it just measure the tablet as it is?
The tablets are measured as they are, yes. There is not further sample preparation that's required, so I don't have it shown here, but let me get to my screen where I do have it, and you should be able to see the tablets here, right? So this is the tablet put into the sample holder. That's the tablet as it is, no additional preparation of that material. And if we were measuring powders, we could measure those in a plastic bag, and we could measure those directly. This can be done with powders, but again, the sample holders are optimized for tablets.
Great, and you've just slightly touched on that. Our next question was what other solid dosage forms can be determined? Powders, premixes. These were the questions that one of the listeners was asking. So I don't know if you want to just expand a little bit more, or do you think that was covered?
Yeah, they can be measured here. Again, depending on the extent of requiring measurement there versus on the solid dosage form, it may be advisable to actually have a system where you're using a fiber optic probe just for those, and giving you more versatility of the different sample forms that you're measuring. So you can measure tablets, solid dosage forms here, and you could put as well powders into bags and measure them as well. But if you are going to be measuring predominantly powder blends and such, I would recommend a different configuration than the Transmission Raman. I would use the fiber optic probe and go directly onto through a plastic bag into the powders.
That makes sense. Next question, asking for some information. Hopefully I get this right. Do you have any references from EP or USP for a customer who's using this for batch release?
There is a publication on the use of Transmission Raman spectroscopy for that. So there are things in the open literature that exist. It has been done with Transmission Raman spectroscopy. Okay,
great, thank you. Is there a sample holder to measure multiple tablets at once?
This instrument, as it is now, is a single tablet holder, but developments into multi-tablet holders are in the works as well for the future. But at this time, this is a dedicated single tablet measurement system. Yes.
Lovely. What is the criterion for selection of the best SEP and SEC value?
What is the criterion? Okay, so that would depend, first of all, on the error of what your methodology is, your reference method and the error of those. So you would not choose a model wherein that would be lower than of your reference value, but it depends on the percent variance of your data that is explained, and how much, by explaining more variance, how much signal you're modeling as opposed to noise you may be modeling. So there are various criteria, and depending on the software one uses, there are automatic choices that it makes, but it is very important that one look at the data, much as we did here, where we looked at making sure that we're understanding what the signal is and how that's relating to what it is that we're measuring. So let me go back to here. So here we have our model. We looked at our model carefully in terms of the error for one thing, but also what signal is being modeled. Does it relate to the components that we are modeling? So there are numerous considerations in that respect. So the error of your reference method, the signal that you're modeling, the percent variance that you're modeling, and also your cross-validation. So by explaining more variance in your data, are you able to predict samples better, or do you introduce more error in your predictions?
Thank you. Our next question, I just want to say thank you so much. We've got lots of participants sending in their questions. So the next one is, how often would you expect to need to recreate your calibration?
Calibrations should be stable over time unless you have changes in your formulations or some of your ingredients. So we don't expect that you would need to recreate calibrations on a regular basis. As with any calibration, some type of regular methodology to check the calibration is always recommended, but the calibrations themselves should have stability over time.
Okay, perfect. Can this technique be used for biomolecule applications, so enzymes or proteins?
Raman spectroscopy can be used in those areas. Keep in mind that Raman spectroscopy is not necessarily a trace level technique. Here we've shown low dose pharmaceutical ingredients at 0.5 weight percent. Depending on your matrix, you may or may not be able to detect lower levels. But yes, Raman spectroscopy is appropriate for many different types of samples. This system uses 785 nanometer excitation laser. If one has biological samples and natural products, it may be recommended to use 1064 nanometer laser, which we do have available in our other instrumentation. But we'd be happy to talk to you about your specific applications.
Lovely, thank you. Catherine, another question that we've had in, if we have two software, are both of these 21 CFR Part 11 compliant?
The BW Analyst software is 21 CFR Part 11 compliant.
Great, I'm sure they'll be happy to hear that. So every time, will it be necessary to perform the calibration model, or is this a one-time activity? I'll see.
The calibration models are meant to be used over time. So yes, it's not, there is not a necessity to recalibrate at regular intervals. As I noted, good practice would be to check the model on a periodic basis to make sure that that's the case. If there is a change of ingredients or change in formulation, then calibration changes would be necessary.
Great. And our next question, how does the amplification cavity affect the likelihood of sample burning, and is this often an issue?
Actually, under this current design, because we are using more defocused light than typically in Raman, where we have a focused light over a very small sample area, sample burning is not seen as an issue. So that's a good question. So you're not reflecting back a highly focused light. You're taking back the reflections from that diffusely reflected light to enhance the signal.
Great. Our next question that we had in, have you ever measured complex active compounds such as botanical extracts in food matrices?
We have measured them for raw material testing. So we have done that using our Nanoran 1064 for incoming raw material. We have not had occasion to do that in food matrices to quantify it with our QT-RAM. But we know that they are Raman active, and they could be measured through Raman spectroscopy, yes.
Okay, great. Clarified that. Kev, do you have an instrument that's suitable for creams or ointment?
Yes, yes. Again, if I go back to showing you the overall picture, which is not our full product line picture, but it does show you the general availability of different instruments that can be used. So with these that are fiber optically coupled, they can be interfaced to cuvette holders, liquid vials, and such. Or if your creams and things are in some other type of container, depending on what the container is, it could be measured directly through that container.
Lovely. Just going through to our next question here. Will it be acceptable to the FDA to release the batch?
Again, that would depend on your validating your methods and showing that, yes, you have a fully validated methodology.
Okay, great. Do you have a universal or adjustable table holder?
tablet holder. The tablet holders that we have, the round one is of a set dimension. Unfortunately, I did not include a picture of the oblong one. The oblong one can accommodate different sample sizes and dimensions. Here, of course, you could have something smaller as long as it's larger than the hole on here. But these can be made, and we do have a set. We do have different dimensions available, but they can also be customized.
Okay, perfect. Is there any comparison of the performance of the transmission Raman mode and the normal Raman regarding this application?
There have been publications in the past on that. Actually, in the past 20 years, there are probably numerous publications in the open scientific literature that one could look at for that.
Great, thank you. We've got a little more time. So what excitation laser wavelength options are available, and are they interchangeable within the same instrument, or do they require separate instruments?
Our instruments are all dedicated to one wavelength excitation, so our instruments do not have a user interchangeable laser, and the QT sampler is dedicated with a 785 nanometer excitation laser.
Great, thank you very much. So I'm going to ask this question. You might need to just correct me a little bit. Why constituent with high fluorescence doesn't mask the spectra of the product?
So if you do have high fluorescence, it will mask the signal. But in this case, if I show you the picture of our spectra here, we do have some fluorescence, but it's not too much to actually be a problem for us. I'm showing here, I will soon have my spectra here. So this background here is fluorescence, so it's not masking the peaks. We still see the peaks that are up here. And in building our calibration models, we applied a first derivative as well as a normalization to our data to minimize these effects. But if we have stronger fluorescence, it can interfere and mask our ability to develop calibration models.
Great, thank you. I think we've got time for maybe a few more. So would you be able to explain how to perform the blend analysis?
Well, you would need to have your samples blends as we have here in this same way, have samples of different concentrations, and to make the measurement of each one of them. In this case, I would do this either with our QT RAM and put it in a plastic bag within the holder, or with one of our other portable systems, preferably with a large spot size, so with our ST-RAM system, and make the measurements, and as in this case, develop calibration models and validate them.
Great, thank you. And what are the challenges during calibration?
The first challenge is making sure that you have samples that sufficiently and correctly span the calibration range that you're interested in, and that you have a reliable reference method. Those are the largest challenges.
Okay, perfect. And have you successfully installed the system in any pharma industry for CU analysis?
This is being evaluated by numerous companies at this time, and we do have an installation at one company that has used it. Again, Transmission Raman spectroscopy itself is a well-recognized technology with growing adoption for this application.
Great, thank you so much, Catherine. That is all that we have time for today. Thank you to the audience for sending in all those questions. If there are any that were unanswered, they will be responded to via email. So if you could please join me in thanking Dr. Catherine Becker for her time today. As you leave the webinar, a survey will appear on your screen asking you to rate the webinar. So if you could please take a moment to provide your feedback, And if now is not a good time, the survey will be sent to you shortly via email. If you can complete this when you can, we would greatly appreciate it. So on behalf of European Pharmaceutical Review and B&W Tech, a Metro Group company, I'd like to thank you all for attending today's webinar, and we hope to see you again soon. Goodbye.